So today's presentation is going to be on signal transduction pathways. Normally, I don't really, other than a few major like Darwin and Mendel and a few Watson and Crick, I'm not really big into the timetable and people, but I always get a kick out of this particular person. This is Earl Sutherland. He won the Nobel Prize for discovering the mechanism for signal transduction. And I always tell students this is Mr. Milne's grandpa. It's really not, but he looks a lot like Mr. Milne's, and he definitely looks like he could have been his grandpa. So it's probably not as funny now, but when I tell kids in class, they think, oh. So he came up with a 3-step process, and it was reception, transduction, and response. So basically what you have going on is you have a molecule, and this is actually the one he discovered, the epinephrine transduction pathway. So you have a molecule, a ligand that's going to bind to a receptor on the surface of the cell. If these are polar molecules, like epinephrine, they have to bind to the surface. If they're non-polar, then they can usually move right through the membrane and bind to a receptor inside, in the cytoplasm. So in this particular case, we get a receptor binds, and then transduction is basically the signal, that's the signal cascade. So in this particular case, we activate something called G protein. G protein goes over and binds with something called adenylyl cyclase. You can see up there. And then that's going to activate cyclic AMP, which is going to activate protein kinase, which is going to cause something to happen. Okay, so we have reception, transduction, and then that ends in a response. In this particular case, the response is to stop the production of glycogen and cause glycogen to get broken down and release sugar into the bloodstream right away. Epinephrine is the fancy name for adrenaline. So this is our fight or flight response when we want to release sugar into the bloodstream right away, so we're ready to fight or run. So here it kind of breaks it down into if it's a polar molecule, it'll bind to a receptor, change the shape of that receptor, start a signal transduction, and then we activate a response. The response is usually activating an enzyme or causing a protein or an enzyme to be produced. So mitosis, the mitosis pathway is one of these signal transduction pathways. So you have growth hormones that can bind to the surface of the cell. Okay, platelet-derived growth hormones do it when there's an injury. Other growth hormones will do it when you're just growing and getting bigger. But basically, in order to repair or grow, we need to make new cells. And so growth hormones will bind to the surface. They will change the shape of this receptor. In this particular case, they cause it to become phosphorylated. Once it's phosphorylated, then it starts a reaction, a chain reaction. So GERB, SOS, RAS, RAF, MEK, and ERK are all proteins in the signal transduction pathway. They carry the message from the signal to the nucleus to trigger mitosis, to trigger cell division to take place. All of these are considered proto-oncogenes. When we get into mitosis later on, all of these particular proteins, if they get modified into a permanently on state, can trigger mitosis to keep happening over and over and over again, and that's what we call cancer. So proto-oncogenes are good genes for regular mitosis. When mitosis is necessary, we need these genes to be activated so that we can do cell division. When proto-oncogenes get modified, then we call them oncogenes. Okay, oncogenes are cancer causers. So if RAS gets modified so that it's turned on permanently, now it's going to get this message going constantly, and we're going to have out of control cell division. Sometimes signals are nonpolar. Okay, the signaling molecule, a lot of our sex hormones, testosterone and progesterone, and they're all the sex hormones are pretty much small steroid hormones. And these small steroid hormones can move right through the cell membrane and bind to, oftentimes, nonpolar receptor molecules that can then move right through the nuclear membrane and actually act as transcription factors. So they can start the transcription of a mRNA, which is going to go out and make a particular protein. So that's a little different, where we're moving through a membrane, binding, moving through another membrane without any issues. The three main types of receptor proteins for polar substances that we see are G protein linked, tyrosine kinase linked, and ion channels. And we'll look at each one of those. So here's a G protein coupled receptor. Basically, you have this G protein receptor that's going to be a transmembrane protein. It's going to have lots of passages through the membrane. I think typically it's 7 passages through the membrane. And there will be a receptor on one end, a binding site, and then there will be a spot where it connects to the G protein on the inside. So when a molecule comes and binds to that site, what happens is the GDP that's on the G protein gets replaced by GTP. That activates the G protein. That causes it to release from the receptor protein and go over and bind to another enzyme nearby in the cell membrane. When it does it, it activates that enzyme. That enzyme often activates cyclic AMP or some other signaling molecule, and a cellular response is going to happen in response to that being activated. This is exactly what's going on with our epinephrine that I showed you earlier. We'll jump back to it for a second. So here we had our G protein, epinephrine activated it, and we had activated G protein, comes over, binds to adenylyl cyclase, activates that, cyclic AMP, blah, 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 and we end up with a response. So another type of receptor is called receptor tyrosine kinase. This one actually takes 2 signal molecules. So this is the one that actually works on with mitosis, and we get 2 molecules of growth hormone that bind to the surface of these proteins. And then when they do that, those 2 will kind of drift in the cell membrane together and form a dimer. When they form a dimer, that activates the tyrosines to become phosphorylated. Okay, so then our tyrosines become phosphorylated. That makes them fully active. They then will activate relay proteins that are going to start the transduction cascade that's going to make its way from the surface of the cell to the nucleus, where it's going to activate some sort of response. And then the last one is an ion-gated channel. Basically what you have is you have a channel that has a closed gate. But when you bring a usually a neurotransmitter, up to bind to that. It opens the gate so that ions can flow in. Okay, so this is an ion channel, and it has a gate that's closed. Okay, and the neurotransmitter opens the gate. There's a higher level of the ion on the outside than the inside, so it's not active transport. It's just a gated channel. As soon as you open the gate, things will move from high concentration to low concentration, and you'll get a cellular response. This is what happens in neurotransmission. Okay, we have neurotransmitters get released. They bind to ion-gated channels, sodium channels on the either muscle cell or the nerve cell. They open those sodium channels. Sodium rushes in. That depolarization of the membrane allows us to start a signal, a nerve impulse moving down the next cell. If it's a muscle, it'll start a nerve impulse moving along the muscle to signal for contraction to take place.